So I'm really happy to be hosting Doctors John Ridenour and Rafal Donczew to discuss their current BMC Methods paper entitled An End-to-End Workflow to Study Newly Synthesized mRNA Following Rapid Protein Depletion in Saccharomyces cerevisiae, and the associated detailed protocol in protocols.io. Rafal got his PhD degree in Poland for a biochemical characterization of DNA replication in an important human pathogen, Helicobacter pylori. A microbiologist by training, he decided to change research topic for his postdoc and joined the lab of Stephen Hahn at the Fred Hutchinson Cancer Center in Seattle. During his postdoc, he studied the fundamental mechanisms of transcription in eukaryotes, applying a range of biochemical and genomic techniques. In 2022, he started his own lab in the Oklahoma Medical Research Foundation in Oklahoma City. He and his group are interested in fundamental mechanisms that regulate and coordinate gene transcription in eukaryotic cells. John received his master's and PhD from the University of Arkansas, where he studied how fungal pathogens recognize and adapt to their host and how gene regulation facilitates these processes. For postdoctoral training, John worked with Michael Freytag at Oregon State University using filamentous fungi as a model to study mechanisms of chromatin-mediated gene silencing. Joined Rafal as a lab staff scientist and is working to understand how chromatin and chromatin-associated factors contribute to the recruitment and activity of core transcriptional machinery. Outside the lab, John loves spending time with family and friends, exploring the outdoors, and finding a great restaurant. John and Rafal, again, thank you so much for being here.
Thank you.
So to kick off,
thank you for having us.
Can either of you briefly explain what the protocol is about, how to use it, and why it is relevant?
So I think I can jump in here. So very briefly, so the protocol allows any user with, we believe, limited experience to quickly get the answer to the following questions. So what is the level of transcription of individual genes in your sample, right? So we use a model organ, Saccharomyces cerevisiae, but we believe that, and we already tested that the protocol can be adapted to other models as well. So the main advantage, we believe, of our protocol is that even though there is no method in it is novel per se, we made it really easily adaptable for anyone. So the protocol describes every step from the growing your culture, labeling RNA, until to library preparation and data analysis, which is usually the big hurdle for many labs out there. So following the protocol as it is published on protocols.io, we really strongly believe that most users won't have much problem to get the answers they needed. And second big advantage is that the protocol bypasses any commercial kits out there, so it's really competitive in pricing. So that's so even if some troubleshooting is required, shouldn't be a big problem for most labs out there.
And you mentioned, Rafal, that even though like every section of the workflow is not novel per se, the protocol completely as described is. What was the motivation for you to put together such a workflow?
So when we started, like, as you said, like the lab is relatively new, so we are obviously trying to streamline many things as much as possible and drop the pricing. But mostly to have the protocol in this shape allows us to easily get the people on board, right? So if we get new people in the lab, it really is a matter of few days for them to become proficient in following this pipeline and to do pretty, I would say, fancy assay, right? Because RNA-seq, for many people, it's still a fancy name. Many people are a little like taken aback by this whole next generation sequencing, but they shouldn't be. This is really easy, and obviously you need to rely on a core which will do the sequencing. But with that, let's say many companies offer that. You can use services at the nearby universities. I really believe that even small labs with limited resources shouldn't have much problems in adapting this protocol for the use. And this is not a normal RNA-seq also, right? So our protocol implements the method called SLAM-seq, which is following the newly synthesized RNA, which gives you a big advantage compared to the classic RNA-seq approaches. Depending on the questions you're asking, right? It was shown in transcriptional field that only the measurements of newly synthesized RNA give you reliable information connecting the perturbation you do with your cells, your culture, and the outcome. So at the same time, the sample allows you to measure both steady state and newly synthesized RNA. So there are a couple of other advantages we may maybe discuss in details later, but I think the big picture is that this method is really like covers it all, we believe. And obviously it has certain limitations compared to other methods, but for this general overview of the global transcriptional state in your cells, I think this is really the ideal approach.
And to
Yeah, and I'll
further
just add,
understand,
oh, sorry, if I can, I would just jump in too to add too that that we also took a kind of a modular approach to building the protocol. Again, based on kind of some established methods. That then we incorporated into like a step-by-step reusable protocol. But the benefit for the modular aspect, the kind of that we think about is that maybe users have a certain preference for library prep or an alternative strategy for different modules. And the module nature allows them to kind of swap out things that may work better for them or that they're more comfortable working with, which we hope we think that will alleviate some limitations to implementing a new protocol.
So, and to help me understand better the significance of the protocol itself, can you describe what was out there already when you decided to engage in this project and why these previous protocols didn't work for you the way that you wanted? So I got that pricing was one of the motivations, and second, the modular approach that you, John, described. So anything else that set this protocol apart from the existing previous protocols?
So I think like the key part of the protocol is this SLAM-seq method, right? A very sort of nice method developed like several years ago and applied, I would say, scarcely. And we believe that one of the reasons is that there wasn't an actual decent protocol out there. There were methods in the papers, and the computational part sounded challenging, but it's not really so challenging as it is. The authors, they provided the software to process the data, but it still requires certain amount of knowledge to get in there. So when we first encountered this, we are using a different approach, similar approach, called 4TU or 4SU RNA-seq, which is also based on the measurement of newly synthesized RNA, but the methodology is much more complicated. And that was the method I applied a lot during my postdoctoral studies, but it was really like, it took its toll, and it was laborsome, pretty pricey, and so on. So when we started here, we had a chat with John, and we thought, okay, let's give it a shot with the other approach, which sounds Sounds complicated, but it doesn't need to be. And we believe that it may actually streamline lots of the problems we encountered with the previous protocol. So, and that turned out to be. So it seems this method is really, in our experience, it's the most efficient and the fastest approach to get the measurement of the newly synthesized RNA in your cells. Although, that being said, if your question is, for example, where polymerase 2 is, active polymerase 2 is located on your genome, that's not the method for that. There are other approaches, very complicated, but they will give you this answer. If your output is the measurement of RNA levels, this is the way to go. So I think answering your question briefly, so the main motivation was mostly that it maybe not that other protocols didn't work for us, but they were pretty laborsome compared to this. And we were looking for something faster and easier to teach also, right? Easier to adapt by a new group, by new people, new trainees. And that turned out to be really the way to go.
And what were the highs and lows in developing the protocol? And an extension of the question is, was there anything that you tried while putting together the pipeline that didn't work and didn't make it to the final version of the protocol?
Yeah, maybe I can jump in here. And I think developing a detailed protocol, definitely takes some time. And so I think, it's kind of a newer lab. In any lab, your time's valuable. So it does take some time, and some of that time, takes away from answering questions that we're interested in. But as Rafal's discussed, there is a lot of value in this. So, I think the time that it took, I would say, is kind of one of the lows. But then, obviously, the best part of developing a protocol is being able to share it. And to see others use it and to see it work for others is kind of the best part. And there were obviously some things that we tried that didn't work out. Just briefly, we had some struggles with kind of optimizing the method that we use for library construction. But then, as Rafal mentioned, we kind of got away from kind of kits and kind of homebrewed our own approach, which is works really well and is robust. And then we also had some hiccups during as we were kind of getting data analysis, our workflow for data analysis set up, but in the end we kind of worked out most of the kinks, or all of the kinks, we think.
And the protocol, when you published the paper, was applied on the yeast Saccharomyces cerevisiae. But you also mentioned briefly that it's being tested in other models. Can you elaborate a little bit on that? And what other models and what key modifications you have to apply to the protocol to make it work in other organisms?
So we have two collaborations going, and so one of our collaborators is applying this protocol for Candida albicans. So that's basically a different fungi out there, and so it doesn't require much modification. We actually, I don't think it will require any. This is still ongoing work. Another collaborator here locally, he applied this protocol for the mammalian cell lines. So the SLAM-seq itself was first developed and tested in mouse embryonic stem cells. So, but again, these are just cells, and this is just DNA and RNA in the end. So we tested our protocol with their cell lines, and it worked pretty well. So certain things need to be adjusted. So for example, the labeling, for labeling, you don't use 40U, you use 4SU. These are the details which are discussed in the protocols. I don't think we need to go as deep here, and the concentration of the labeling compound needs to be adjusted. There is enough literature out there which allowed, it wasn't really a big problem. And there are a couple of examples you can find in the papers. But other than that, the library, the data processing, basically it stays the same. You just change the reference genome and you can get your output for the human cell lines, for example. So we really believe that this protocol can be very helpful for not only the Saccharomyces cerevisiae labs, but any lab out there.
And I want to change gears a little bit now and focus on the motivations that you had to share your protocol on a public repository and have it peer-reviewed and published as a protocol paper in BMC Methods. What were those?
Yes, so we definitely wanted to make this protocol as accessible as possible. So we already had our interactions with protocols.io before, but maybe never so formal, and we never did such a, I would say, thorough job with depositing the protocol. But we like this. We like the layout of that. So that's definitely, it's much easier for people to follow the protocol in protocols.io compared to the typical methods section in the paper, right? Which are very cramped and sometimes some details are missing. So we wanted to make it very accessible. At the same time, we wanted this protocol to go through the actual peer review process to give people some, piece of heart that they are following something that's, that was reviewed by the experts in the field. So I think the combination of these two approaches really convince us to go ahead and publish this in BMC Methods and deposit the protocols in protocols.io. So we'll have the best of two worlds.
And what do you think are the benefits for you of BMC Methods' transparent peer review system, where the reviewer reports are published online alongside the article?
Oh yeah, so I think some of the benefits, of publishing having the reviewer reports published alongside the paper. I think that those, kind of as Rafal mentioned, we can, kind of see what, some of the experts or other researchers, thought of this work, and and maybe some additional insight into the methods that, kind of based on the constructive feedback that's in the reviewers' reports. I also think it's important to see, how that constructive feedback, does improve the final product, the final publication.
And the final question that I have is, what has been the significance of this protocol for your research team and individually?
So since we, as John mentioned, we spent a while developing this, and we are kind of on the fence, thinking, okay, is it really worth it? But it turned out to be, because we are getting quite a range of trainees in the lab. Some people coming here just for a few months, working in the summer and so on, or maybe just temporarily for several months. For this setup, it's extremely helpful to have something so well established, right? So we just give them the protocol with minimal training for a few days. They can really do these experiments, so for others, it may sound very complicated for a trainee, but in our case, it's not it's really easy to get it working, because the protocol is I believe it's very resilient to small errors. There are a couple of steps we are omitting from the typical RNA-seq approaches, which are discussed in the BMC Methods paper. So with these modifications, this protocol is really resilient for new researchers or not very well-trained researchers out there. So that made our life much easier. Now, for example, another good example of how this protocol is helping in the lab is that we are moving a little towards a new direction. We are thinking a little about how transcription and DNA replication interface looks like in the cells, how these conflicts are resolved. So this method we already tested this initially, that this method will allow us to, rapidly track the transcriptional changes across the different, let's say, cell cycle progression steps. And the reason for that is that this protocol, the way we develop it, it's very time sensitive. So really whole labeling and initial RNA processing takes several minutes. So you can get basically your resolution in time is limited to several minutes, which is just a tiny fraction of the length of the cell cycle in cerevisiae. And so that it's also true for other models. So I think with having that, you can get this very detailed snapshots of what's happening in the cell at different stages of the cell cycle, if that's the question you're asking. And for other approaches, if you are not bothered by the cell cycle, you just want to get the general outcome, this is also working very well. So I think it's really, the protocol is very, I would say, flexible, and it can facilitate many different approaches and experiment and questions being asked.
And so I don't have any more questions. I don't know if you want to mention something that we haven't touched so far.
I think I would just add that for anyone listening, that if you guys have problems or run into any hurdles, adapting this protocol, just feel free to reach out to us. We'll be more than happy to help and to answer any questions you may have. And we really believe that we can make it work for anyone.
Since you mentioned troubleshooting, I'm going to ask a question. What are the most critical steps for the protocol? Those where users have to be extremely careful.
I think John will have a good answer to that.
I think kind of, one thing that Rafal did allude to was the timing. And so I think when we think about this protocol, we think about the time is a key aspect. Particularly during protein degradation in the 4TU incorporation. But with one thing that we kind of worked out was nice guideline for using rapid methanol fixation to kind of alleviate some of the strain that we find trying to keep everything on time. So that's we kind of think about that as kind of a critical step. One other thing that we should mention is that the following the 4TU incorporation, alkylation of the modified RNA is a key step to the SLAM-seq protocol, and the alkylation is done by using a reagent called iodoacetamide, and that's a relatively unstable compound. So we have some recommendations in the protocol to make sure that that we're trying to prevent that degradation or instability as much as we can.
Well, Rafal and John, thank you so much for your time. It's been insightful, and I'm sure that the information you have shared will be of use to the readers and users of the protocol.
Thank you very much.
Yeah, thank you again. Yeah, thank you.